Hello and welcome to PW English channel, and I am your Diksha Sharma ma'am. So today we are going to start with our chapter Structural Organization in Animals, wherein we are going to discuss the animal tissues. Alright guys, so this chapter is really very important from the NEET point of view because this chapter actually contains the weightage of around 2 to 3 questions, one or two questions from the animal tissues part, and the second one is from your favorite animal, that is cockroach, and that comes under the animal morphology. So today of this chapter, structural organization in animals, we will be covering only the tissue part, right? So let's get started. So here, what is the tissue first of all? So all of you must have heard or you have learned the definition in your lower grades. Like tissue is a group of cell. But does it really just a group of cells? No, but here because you are studying more of the concept, more deeply of the concept. So let's just discuss what actually tissue is. Tissue is a group of cell, and that cells, they are similar in structure, function, and origin, right? So whenever we are saying the word tissues, we are talking about the group of cells which are similar looking, which are performing the same function. For example, if I say your muscle tissue, all the cells in a muscle tissue, they are of the same shape, same size, they appear same, right? They're similar looking, and also the function they are providing or they're performing is same, like muscle contraction. They are not doing any other function. They're not protecting your body. They are what? They're not covering your body, right? They're performing the function of what? The muscle contraction. So herein comes the concept that what is a tissue. We just not say a group of cell is a tissue, because group of cell may be, it can be a cluster of different cells. For example, a muscle cell, a neuron, an epithelial cell, right? We have three type of cells. So if there is a cluster of all these three type of cell, you will not call it as a tissue, you will call it a cellular aggregate, which is seen in your phylum Porifera. So the first phylum to have tissue, your Cilientrata, where actually the tissues are formed, we say the group of cell which is similar in structure, function, and origin. And second part of this definition is along with their intercellular substance or matrix. So in between the cell, there are a lot of substances present which are actually of the utmost importance. For example, if I say I have two cells, if I have two cell and these cells, they have fibers in between. Fibers helps them to communicate. So you just cannot ignore them, right? Because they're performing a very important function. So here comes the complete definition of tissue. You're learning a full-fledged definition. And what is that? The group of cell, group of the group of cells similar in structure. Structure, that means it appears same. Function, that means it performs the same function. And what about the third term, origin? And origin along with its intercellular matrix. Along with its intercellular matrix. So what's about the origin? When you were embryo, when you were embryo, you your cells, the initial stage cells, they all were the totipotent, they were like stem cells. Later on, the differentiation occurred in the cell, and that cells, they get differentiated three germ layers, and that germ layers are ectoderm and endoderm. Later on, all these germ layers give rise to all the body parts, right? For example, if I say your muscle is right, this is how we say that we actually originate from all these three germ layers that are ectoderm, mesoderm, and endoderm, right? All right. So if I'm talking about the zoology, not about the botany, I'm just talking about zoology. There must be some person who have coined the, for example, when you were born, someone in your family must have given you a name, right? Similarly, the scientist named Bicart, Bicart coined the term in zoology, coined the term, right? So the study of tissues. Now, what the study of tissues is known as? It is known as histology. Histos means meshwork, because there is a meshwork of the fiber-like things. So it appears like a meshwork of something. They gave the term histose. Histose means something with fiber like and meshy thing. So here that's the term histose. So logy means studies. From there the term arises, which is the study of tissues. All right, moving further to the next, as we have discussed, which is the first phylum in the animal kingdom to have tissue. So you will be learning a lot of phylums in the animal kingdom, right? The phylum starts from Porifera and ends up to Chordata. So we consider that phylum Porifera is the simplest one, right? And the complex one is the Chordata. The simplest organisms are poriferan sponges, they are also sponges, and they do not have true tissues because they are simplest one. With evolution, when the organism advance, the cellular aggregate get turned into, with the cells they get specialized, they are similar looking, they're performing the similar function. That cluster. So the question arises, which is the first phylum to have tissue? That's ciliate. So first phylum to have tissue, first phylum have tissue, ciliate. In fact, this is also the first phylum to have sensory cells and a nerve cells because uh, all right, to so I have written ciliate. The first phylum to have tissue, the ciliate. All right. So this is also the first phylum to have the nerve cells and the sensory cells because poriferans, they lack these sensory and nerve. Also, they lack the tissue level of organization. So everything which is more advanced, it started for the first time in the phylum Ciliate. All right. Okay. So this was about the introduction part. Let's see what type of tissues do we have. Right. As I've told you, one is the musculus. I've given you one example. I've told you one clue, right? So now it's your turn to guess the other ones. Okay, I say there are four types of tissue. Tell me what the four types of tissues are. One is epithelial, second is connective, and third is muscular, and fourth is nervous. So let's write them down. So if I say according to its location, we have classified them according to location, structure, and function, right? So there must be a basis of classification, right? You just cannot classify it blindly. The basis of classification is where are they present, how they look like, and what the function they. So we have four type of tissues: epithelial tissue, connective, muscular, and the nervous. Fine, guys. So here, all right. See everyone, attention, pay attention. So very crucial class. If you have done all of this, like if you have seen this class or have you learned a lot of things from this class, it will be pretty easy for you to crack the questions. So I would like to like all of you to pay attention here and also to put good notes because without notes the study is incomplete. So please, I suggest all of you to have a copy or a notebook handy with you along with the beautiful colored pen so that you can write it because more colorful the copies, more likely get attention, you pay attention, more likely you have to have a pictographic or photographic memory and you remind those things and that gets imprinted in your memory for a longer time, right? Okay, so first we have epithelial tissue. The epithelial tissue, what is its origin? So as I've told you, all your tissues have originated from the three germ layers. What were the three germ layers? The ectoderm, mesoderm, and the endoderm. So which germ layer, from which germ layers epithelial tissue have arrived? The epithelial tissue have arrived from all the three germ layers. Yes, that's ectoderm, mesoderm, and endoderm, right? So its origin is from all the three germ layers: ectoderm, mesoderm, and endoderm. Talking about the connective and the muscular tissue, their origin is from mesoderm. Their origin is from mesoderm. And this one, nervous tissue, its origin is ectodermal with one exception. And what is that exception? Microglial cell. Now, what are these microglial cells? We'll be discussing it in the nervous tissue. So, what is the origin of microglial cells? Otherwise, the entire nervous tissue has been originated from the layer ectoderm. All right. So, please write this down in your copy. There are four tissues: connective, and nervous. All right. Moving further, epithelial tissue. Let's talk about them. But before talking about the features directly, I would like to draw a diagram. I will also please draw the diagram with. And then side by side, we'll be writing all its. That will be very simple. All right. So let's have your colored pens handy and draw the epithelium. So if I'm saying the cluster of the cells which are similar looking, performing similar function from similar origin, call it as a tissue. So for an instance, if I say these are epithelial cells, the what? These are right? So that's epithelial tissue. Epithelial tissue, it just cannot live alone. For something, for like support or strength, it must be needing something here below it, right? So beneath these epithelial cell is present a basement membrane. What it is present here? Here is present basement membrane, which is made up of two layers. One is the layer which contains the fibers and the another contains the glycoproteins. And this entire layer, you call it as the basement. So the basement membrane is made up of two layers. One is the basal lamina, the one in the basal lamina, just made up of glycoproteins, and the one below it, this one, the reticular lamina. The reticular lamina contains fibers. Contains fibers. The main function of the basement membrane is to support the epithelial cells and to provide strength. And to provide, right? So the basal lamina, it is made up of glycoprotein, right? And mucopolysaccharide. What are mucopolysaccharide? You will learn it in the biomolecule, right? These are like carbs which are slimy, slippery, like when you eat. If you have ever cut down the okra in your home and you have helped your mother in cutting the vegetables, you must be knowing that the okra, when you cut it, it's little greasy. And ask your mother to help you thing, right? It is that slimy, or you have ever seen that in the aloe vera, right? So that slimy thing is your mucopolysaccharide. So here the reticular lamina is made up of fibers. Beneath this basement membrane is present the connective. This connective tissue, it contains blood vessels. What does it contain? It contains blood vessels, right? Connective. This connective tissue is the another tissue, second tissue. Here I have drawn the two. One is epithelial, another is. So let's talk about their features. First of all, if you can see here, you can the cells, they are very tightly packed. They are just present, they're just lying next to each other. There is no space in. So we say that the epithelial cells, they are tightly packed. Epithelial cells are tightly packed with no or less, with no or less intercellular matrix, or right. So what is the intercellular matrix? The matrix present in between the cells which contains fibers that we have talked about, right? So the this intercellular matrix is very less because the reason is the cells are tightly packed and the space between them is also less. Fibers will early, right? So here next, if we can see this, there is just a single layer present cells, right? There can be a possibility there are a lot of other cells may be present above it in the multiple, right? So you say that the epithelial cells, they can present in the single layer, they can also be present in the form of multiple layers. There can be a single layer and there it can be of multiple. The cells are present, the form of single layer or multiple layer. So whenever there is a single layer of epithelial cells, you call that epithelial tissue as simple epithelium. And when there is multiple layer, you call it as compound epithelium. Right? So this is how we have classified the epithelial tissues. One is the simple epithelium, another is the compound epithelium. There is a multiple layers, it's compound. If there is a single layer and that is directly attached to the base, simple epithelium. Moving further to the next feature, you have noticed there is no blood supply in the epithelial tissue. And you will say, ma'am, yes, there is a blood vessel. But this blood vessel is present in the connective tissue. It's not present in. Can you see that? In epithelial tissue, there is no blood supply. The blood supply is of the connective tissue. Say that epithelial tissue is avascular. Avascular means no blood supply. So if there is no blood supply, from where it is going to take all its nutrients, or from where it is going to take its oxygen, and from where it is going to release the oxygen, right? So the oxygen is directly coming into the cell via diffusion. Also, the nutrients are coming to cells in the same process, no diffusion, right? Since there is no blood supply in the epithelial cell, so the blood vessel of the connective is going to give its all nutrients epithelial cells via diffusion. All right. Next point. It also have a nerve supply. It also have a nerve supply. That's why if someone give you a tight slap on the face, you actually feels it. Yes, you feel it or not, because there are open nerves present here, and someone put a pressure on it, they get. And you feel like crying, or you get angry or something like that, because we have nerves down there in the. Next, if you have ever noticed these epithelial cells, where are they present? They're usually present in the form of coverings and linings. Now, what is this? Coverings and linings. You know what is a covering? Whenever you are covering, for example, you have a gift, right? You are going to pack it from right as well as from inside. So if you're packing it from the inside, you will call it as a lining. And whenever you give gift wrapping it from the outside, you are calling it as a covering. For example, my skin is made up of epithelial cells. It's covering. Similarly, I have epithelial cells in the stomach. That will be the lining. Both these coverings and linings, they are in the direct contact from the external, the outside things. For example, this layer is in contact with everything. For example, sometimes it gets rubbed with this board, or sometimes some animal or small kids scratch it out. So the wear and tear is more. Similarly with the linings, the food get entered into your esophagus, and there again there is a crushing of food. The wear and tear of the epithelium over here. So the nature has provided a alternative to what? If you are at that point where you can break things, or you are at the verge of break, the nature given power of regeneration. For example, I give you a very simple example. You all must have taken the bath in the morning, right? So while you are bathing, you slow off a lot of cells from you, right? You slow off a lot of cells from you. But have you ever seen a muscle down there or a connective tissue down there? No. Why? Because the more cells you're destroying, the more than that are also formed at that point, right? So we say cells, they have a power of reach. They can divide, and they can divide and increase their number. Fine. So we say, where are they present? The epithelial tissue is present in the form of epithelial tissue is present form of form of coverings and linings. Covering, so I'm putting examples. You can easily understood it after you write it down or after so. And lining, linings we have like stomach. And also it has the power of regeneration. It can divide, get re. Alright, that's the basic features of your epithelial tissue. Let's move further and talk about the classification part of the epithelial tissue. Moving further to the epithelial tissue classification, as I have told you already, you have divided the tissues. One is the simple epithelium because it has one layer of cell, and then we have the compound epithelium because it has two layer of cell or multiple layer. All right. So we have divided this simple epithelium on the basis of shape of the cell. On what? On the basis of shape of the cell. On the basis shape of the cell. So what type of simple epithelium have? Let's get started. The first one, where the cells are entirely flat cells, you call it as squamous. And where the cells are cube-like, you call it as cuboidal. Where the cells are tall, they are columnar. And where the cells have cilia, they are ciliated. Now, if the cells have cilia, what type of shape they have? If the cells are tall, that's columnar ciliated. If the cells are cube, like it's cuboidal ciliated. And then we have a special another category that is pseudostratified. Not mentioned in CRT, but we'll be covering it. Right? Moving further to the next, the compound epithelium. What about the compound epithelium? So we have divided the compound epithelium into two. One is the stratified and another is the transitional. The transitional epithelium is epithelium you will find in the urinary system. Where will you find it? The urinary. Alright. The transitional epithelium. Where will you find the transitional epithelium? You will find it in urinary system. Whereas the stratified means layer. So it has multiple layers. So you have also classified it further. Squamous, cuboidal, columnar, and columnar cilia. We will be discussing all of this. Talking about the squamous, we have two types of squamous, keratinized and another is non-keratinized. I will suggest all of you write this down in your copy so that we can hurry up. That's important. That will, if you make this chart, it will be pretty clear for you. All right, write this down. Okay, let's move further. You can take a screenshot. Next is the simple tissue. Simple tissue means we are talking about simple epithelium. Fine. So the first category is squamous. If the squamous cells are flat cells, also written. So whenever you are characterizing or classifying all of these categories, the reason or the cells which we are taking these cells, not right. So for example, if you are just looking at a tissue, right, here I can see the cells which are of different sizes, and here I can see a flat, right. So my criteria of classification of is not from this view. My classification is from this view. All right, so I'm not looking at these cells for the classification of. Whenever you are seeing from the vertical view or the side view, you can see the cells are flattened. But otherwise, in any epithelium you're looking at the border, you are looking at the columnar, they all will appear polygonal from the upper views. The polygonal, right? Polygonal means it has different shapes. So if I'm talking about the squamous epithelium, as you can see, the cells are flat. Cells are flat. And also, if you see it carefully, all right. Okay, so if you see it carefully, the cells are flat, plus the cells have irregular boundary. What do they have? They have irregular boundaries. The cells are flat with irregular boundary. All right. Also, if you can actually imagine, these cells are very so these cells are flat and they're forming a very thin layer. So they will be present all those where actually form a function of diffuse or the exchange of cells, right? So if the cells are flat, they will be forming a very thin epithelium, and thin epithelium is best for. If it would be thick, the exchange and very difficult. So it will only be present way on here have to perform the function of or. So where are we performing the function of diffusion and exchange? Exchange of gases between the alveoli and the blood vessels. So both the blood vessel and the alveoli will be having the squamous epithelium. Second, we are also making filtration brain in the kidney. In kidney, we are allowing the filtration of blood to form urine. So here also the things are passing down. So we need a membrane, right? And that membrane will be squamous epithelium, right? So here we are talking about the examples of the location where you will find this epithelium. Since it is thin, it is thin epithelium, so it will be present, will get it in the areas where diffuse or exchange is occurring of substances. This can be air or gases or any nutrients or something, right? So example of the location, talk about them. We have alveoli present in the lungs, have blood vessel, then Bowman's capsule. These are the locations where you can find the squamous. Moving further to the next category. Next, we have cuboidal. You can see the cells, they are cube-like. The cells are cube, and somewhere the cells make microvilli. Now, what is the microvilli? For example, if this is the cuboidal cell, the cell's plasma membrane get extended and forms finger-like projection. Call it as microvilli, right? What is a microvilli? The finger-like projections of the cell. So wherever the microvilli is present, will be increasing the surface area. Okay? The surface area. So like I have told you, the squamous epithelium is present on where you're performing the function of change of substances. Similarly, this will be present in those areas where we are sick, right? We are absorbing something or we are secreting. So where it will be present? Areas of and absorption. Okay. Since this is increasing the surface area, so that's why microvilli is present on absorbing something or where you are secreting something. For example, if I say the microvilli is present or PCT, the proximal convoluted tubule, right? So wherever the microvilli is present, that epithelium is directly known as the brush bordered epithelium, right? So wherever you will find the word brush bordered, it means that that epithelium have the microvilli in it. Wherever microvilli is present, that epithelium is brush bordered. So brush bordered, if I'm talking about brush bordered cuboidal simple epithelium, that means the cuboidal cells, the epithelium is simple because it is single layer of cells. Which type of cells? Cuboidal cells. And if I have added the word brush border, that means those cells will be having the as well. So here, what is the location then? Let's talk about the location. First, as I've told you, PCT. What is PCT? Proximal convoluted tubule. If you don't know what is a PCT, go and see the diagram of a nephron. The nephron is a structural and functional kidney and it contains tubules, and one of the tubules is PCT. You can see it in the chapter excretory products and CRT as well. So this PCT has this actually, this is also a PYQ previous year question. I have asked about what type of does PCT have, or they may ask the question, PCT is it is lined by brush bordered because it has microvilli, brush bordered simple cuboidal. Otherwise, it is also present in gonads and smaller salivary and pancreatic ducts. Smaller salivary and pancreatic ducts. I hope you must have heard the name of a salivary gland that produces saliva in your buccal cavity. right? So the smaller ducts of this gland and in fact of the pancreas contains this simple cuboidal epithelium, but in these two cases it's not brush bordered. Brush bordered is only in the P. Fine. So that's about the simple cuboidal. Let's talk about the columnar. If I'm talking about the columnar, the cells are tall and slender. Cells are tall and also they have microvilli. They have microvilli, so it can be can also be brush bordered. Also, it is performing the function of secretion, performing the function of secretion. And all right, so where do you find it? You will find it in the glands. Which glands? Intestinal gland. Intestinal glands. And even in the glands of stomach. And the glands of the stomach are gastric glands. All right. So wherever the word gaster appears, that is stomach, because the another name of the gaster. Otherwise, you can one very special cell here, goblet cell. Which cell? Goblet cell. So what is a goblet cell? For example, these are columnar cells. So these columnar cells, and they may have a microvilli, right? And beneath this, they have a basement membrane. And they also contain a very special cell, which is a goblet. Goblet cell is a unicellular gland. What is it? It's a unicellular gland. It secretes mucus. That's why you also call it as a mucus cell, the unicellular gland. Your glands can be multicellular. Your glands can. I will be talking in the glandular. This glandular epithelium is a specialized epithelium in which cells have become special and now this, right? So here, if I talk about this columnar present in the glands, and in that glands, because mucus is also secreted there, so they have a special cellular gland, goblet cell. They are producing. All right. So this is about the columnar epithelium. Moving further, the ciliated one. So as I've told you, the ciliated having the cilia contains cilia. Now, what's the difference between microvilli and cilia? Microvilli is the extension of plasma membrane, finger-like, right? And it is for the absorption of the. Whereas cilia, it's hair-like, fine hair-like. It has motor down there, right? You will see that in the cell chapter. And that cilia function surface area or absorption is rather helping in the movement of substance. It's just like this. It's like hair helping the substances. It's just like grass, right? Like grass is moving when there is windy day. Just like that, the trachea is moving. So we have two types of cells. So that's why you have two type of cells. One is columnar ciliated, where the cells will be columnar, the tall cells, and then we have the cuboidal epithelium, where the cells will be. So where do you find it? You will find it in larger bronchioles. What are bronchioles? The bronchioles are the part of respiratory lungs. Like you have trachea divided into bronchi, and bronchi later divided into bronchioles. The bronchioles are of two types. One are the bigger one, and the bigger one then will open into smaller one. The smaller one will be having ciliated, whereas the bigger one will be having the columnar ciliate. Talking about the another example in the columnar ciliate, have fallopian. We have fallopian. Now, what is the function of the cilia here? If I'm talking about the bronchioles, here it helps in movement of mucus. Helps in movement of mucus. For example, for example, there is a dust particles of microbes that enter into my respiratory tract, right? It enters to the bronchioles, and in bronchia, the mucus will trap all these particles and the cilia will propel it out, right? The movement is in a specific direction so that everything gets moved. Also, the fallopian tubes helps in movement of egg or ova, right? Of ova. Fallopian tube is a part of a female reproductive system. We'll study reproduction, and this helps in the of fine. All right. So ciliated. Next, we have pseudo stratified. What is pseudo? pseudo means false, and stratified means layers. These cells, they are actually single layered, but they appear like a multi-layer. How do they appear? They appear like a multi-layer. For example, if I have a basement membrane, right? And these are the cells, tall cells. You will get two type of cells. Both these cells, they are attached to the basement like this, right? These cells, they are apical cells because they are tall, and these are basal. Both are attached to the base membrane, so both are forming the single layer only, but it appears multi-layer if you see the microscope, because you will find two nuclei, right? So that's why you call it as false layer, false pseudo. Right, means so stratified means false layer. That's why it comes into the category of simple, because the layer is single only. So this pseudo stratified is of two types, right? The pseudo stratified is of two types. What's the category? One, we have pseudo stratified columnar cilia, and then we have pseudo-stratified columnar. There are two categories here. So there are two categories here. The pseudo-stratified, one is pseudo-stratified columnar, another is the pseudo-stratified columnar, right? So let's see where are they present. So if I'm talk about the columnar ciliated, right? So I'm writing PS. That means you will find it in the trachea. Where will you find it? You will find it in the trachea. If I'm talking about pseudo-stratified only columnar, non-ciliated, that means there is no cilia. You will find it in the parotid gland, you will find it in male urethra, right? You will also find it in the epididymis. All right. So that's about the simple epithelium. So in simple epithelium, we have done squamous, cuboidal, columnar, pseudo-stratified, ciliated. Then moving further to the compound epithelium, as we have discussed, the compound epithelium is of two types. So first we'll talk about the stratified one. Stratified. Stratified means layers. That means layers. All right. So this stratified one, layers, it is of different categories. For example, we have the squamous, then we have the cuboidal, and then we have the columnar, and we have the columnar. All right. So we'll be talking about the squamous and squamous. Okay. So first of all, they are multi-layered. So what will be the criteria of the classification? Because there are a number of cells. So which cells, what type of cells you're taking as a criteria for classification? The upper layer of. So if the uppermost layer of the cell, they are flat, that will be squamous. Uppermost they are cube, like that will be cuboidal. The uppermost columnar, that's columnar, or the tall cells. And if it's columnar cilia, that means tall cells with cilia, then columnar. All right. So here, if I'm talking about squamous, squamous is of two categories. One is the, all right, one is the keratinized. That means the cell will be having the keratin. Another, the cell will not be having keratin. Now, what is this keratin? Okay, keratin is a protein, and you will find this your your hair, right? So this keratinized, you will find it in the skin, and this one you will find the pharynx, buccal cavity, conjunctiva, vagina, and many more. Okay, conjunctiva is the outermost covering of the eye. It's a protective covering. Vagina is a female reproductive organ, and buccal cavity is mouth, the area inside your mouth, and pharynx is the place after the buccal cavity where the ovid swab is taken, that part. All right. So if I'm talking about how does it look, okay, let's draw this. Let's draw this. If I say this is the basement, and above the basement membrane is a layer of cells which forms a germinal layers, because from here all the cells originate. You call this layer as stratum because also from this layer you will from this layer is attached to the membrane, so all the nutrition will be derived from here directly. Above this is present the topmost layer, that's is squamous, and in between the topmost layer and the basal layer you will see a number of polygonal cells with a lot of side. Okay, so as I've told you, this is the uppermost layer, so that's the criteria of classification. Cells are flat, thus you are calling it as a squamous epithelium. So we have polygon. All right, guys. Okay. So here we are drawing a lot of cells. All right, so these are cells. So I'm drawing all the nucleus down here. Okay. Okay. So the uppermost layer is squamous. So there will be a lot of flat cells. And there will be the uppermost dead layer. There will be uppermost dead layer. And that dead layer contains a protein keratin. Okay. So this layer which contains the keratin plus the dead cell, you call it as stratum corneum. Corneum which is avascular, which is dead. So wherever word the cornea, corneum comes, that means that layer is dead or the avascular, like your cornea and the eye. It is also avascular. So this contains dead cells plus keratin. Now, what's the function of this protein? This keratin, it makes your skin waterproof. Just try it once. Put a drop on your skin. You will see the drop will always remain. That will no side. That's why while bathing, you will have water inside your body, right? And then it also helps in protection. For example, if you have a pet in your house or a very small child, they attempt to scratch your skin. And not every time you get the blood ooze out because you have a dead layer of keratin that gets scratched out and blood oozing out. That means the scratching has been done only up to the upper layer of your epithelium. All right. Then this layer, as I've told you, is stratum germinate. So this, if the squamous epithelium contains the squamous keratin, that will be keratinized. If it does not contain the keratin, it. All right. Okay. Moving back again, if the upper layer of cells are cuboidal, like that will be the cuboidal only. And where do you find the cuboidal one? You will find the cuboidal in the larger salivary and pancreas. So if I'm talking about the smaller salivary and pancreatic ducts, that's simple cuboidal. If I'm talking about the larger salivary and pancreatic ducts, that's the cuboidal stratified. Then we have the columnar. Where do you find the columnar? In the ducts of mammary. You know what is a mammary glands? The breast, the milk producing glands, the female body. Their duct is, it contains columnar epithelium, whereas columnar ciliated present in the larynx. I hope that's pretty clear. You just need to remind the locations in these one. Otherwise, here you have to also have to learn what the keratinized. Keratinized means the keratin is non-keratinous. Non-keratinized means the keratin is absent. And what is a keratin? It's a protein. And where do you find this protein? In your skin, in your nail, in your hair, right? Okay. So this was about the squamous stratified. Let's talk about the transitional. Transitional epithelium is epithelium. When you stretch it, cells can change shape. And this epithelium is actually present in all those areas where the occurs most of the time. For example, your urinary system. Because in the urinary system, what is happening there? The urine formation occurs, and there is also the storage of urine. So all those organs which from which the urine is passing out, they need to be out sometime. That's why nature has given a very flexible epithelium. That's transitional. Because it is present in the urinary system, we also call it as a, right. It is also known as the urothelium. So as, all right. So here the cells change their shape on stretching. Cells change their shape on stretching. All right. Okay, so where do you find it? You will find it in the renal calyces, renal pelvis, ureters, urinary bladder, urethra. All right. So, what are all these organs? If you remember the structure of kidney, the kidney is divided into these pyramids. Okay. And these pyramids, they open into small ducts. What do you call it as? Renal calyces, the minor calyx, which then open into the major. This area is the renal. Okay. So, these are the, and the plural is calyces and the singular is calyx. L Y. All right, guys. Okay, so that's the urothelium. So remember, where do you find the the urinary system? That's right, it's uro, and that is a transitional. Next, glandular epithelium. As I've told you, the glandular epithelium is a kind of a specialized epithelium. This specialized epithelium, it has been specialized in a way that the cuboidal and usually columnar cell, they are performing a special function. And what is that function? The function of secreting something. And whenever something is secreting, whenever a cell is secreting, and you usually call it as a gland. Now that gland can be made up of a single cell like your goblet cell. So that goblet cell is a unicellular gland. Otherwise, it can also be made up of a number of cells and it's just producing a separate organ like your saliva. Fine. What is a glandular epithelium? What it contains? Glandular epithelium contains specialized cuboidal or columnar, cuboidal or columnar cells that secrete some, that secrete. All right. This is how we say the glands. The glands, it can be the unicellular. That means it is made up of a single cell, and it can be a multicellular. What does it mean? It means it contains or it is made up of number of cells. Up of number of. What is this? Like your salivary gland. Also, it can be unicellular. That means made up of one cell only. Now, what's that? Your goblet. Because the goblet cell was secreting mucus, it is also known as the mucus cell. All right. Let's talk about the another classification of plants. Okay, so that's the green. Want the green one? We'll use the black one. Let's just, anyways. Okay. So here the another classification of glands. If I'm talking about glands, I'm talking about the multicellular glands. Glands I'm talking about the, if I'm talking about the multicellular gland. Now multicellular gland, they must be something. And now how this out of that particular gland, all the glands have different criteria. For example, some glands have ducts from which it pour out all its, and some glands that do not have the duct from which, so from where it is going to pour out the they're going to help of a body fluid. Similarly, we have classified the glands into three categories. One is the exocrine gland, another is the endocrine gland, and the third one is heterocrine. What's the difference? Okay, I'm talking about the exocrine glands. These glands, they are situated in or somehow like in your buccal cavity and most of the places, right? They need to put or they need to pour out this. How they're going to pour out this? They're going to pour out with the help of a tube-like structure, and these are here known as ducts. So these glands, they have ducts. Say they have ducts. So for example, if this is your skin, this is your skin, and this is your oil gland I'm making here beneath this. Okay, this oil gland is producing oil, so it has to pour out the oil, the skin with the help of this duct, right? So this becomes the gland, exocrine. So it is pouring out everything it has through a duct. But in the endocrine, we don't have duct, no ducts or ducts absent. So now, how it is going to throw out its secretion? For example, if I have a gland here, the endocrine, right? It is secreting something, and this something here is known as hormones. What do you call it as? Hormone. So its secretion is hormones. Now these hormones, they're going to enter into the blood vessel, and this blood vessel is going into the target organ. So if they don't have duct, they must be needing some medium. They must be needing some medium, and that medium is blood. All right. So the hormones, these endocrine glands have ducts. So these hormones, they are transported towards the target organ with the help of your body fluid. And what is that body fluid? That is blood. So blood helps in transport of secretion. And what is a secretion? That is hormone. So you know a lot of endocrine glands lately, right? What are those? Like your thyroid gland, like your pituitary. Just writing. And the last is heterocrine. Hetero means mixture, crine means secretion. So that's a mixture of two glands. It will be containing endocrine part, and it will also be containing the exocrine part. For example, I'm talking about the pancreas. I'm talking about the pancreas. Let's write the example. The pancreas. And second example is your gonads. What are gonads? The testes and ovaries. These both contain both the parts. One is the endocrine, another is the exocrine. For example, I say the pancreas is producing insulin. That is its endocrine part. That's the hormone, and that will be transported to the target organ, that is liver, through the blood. And if I'm talking about the another part, which is exocrine, that is the enzymes, right? It also secretes, the pancreas also secretes a digestive enzyme. It is moving into your duodenum through the duct. That's your exocrine part. Similarly, in the testes and ovaries, what is the thing? For example, if I'm talking about the exocrine part, the gametes, for example, the ova moves outside through a tube known as fallopian tube, and in the male there is a special duct system, vas efferent, right? If I'm talking about the endocrine part, then hormones again, estrogen, progesterone in the females, and in the males testosterone. So that comes the third category, which is heterocrine. But in this chapter NCRT, only these two categories are given. This is not given, but this will be very useful when you will coordination. All right, guys. Okay, moving further to the next. Next is the cell junction. Cell junctions are the characteristic of your epithelial, yes, of the epithelial. So here, what are cell junctions? These are the point of communication between cells. For example, you are living in the area with your neighborhood, right? Yes. So can you live without talking to? Cannot be. Survive in this society, in this colony. Sometimes you may need some help. Similarly, the cells, they're tightly packed in a, they also need to talk to their neighbors, the neighboring cell, right? So they have a point of communication, have a point of contacts, and are known as the cell junctions. What are cell junctions? These are point of communication, point of communication between. All right. So what are the three types of cell junctions? There are three types of cell junctions. One is the tight junction. Second is the gap junction, and third is the adhering junction. Now, what's the difference? And their difference is based upon the function. Example, let's draw. If I'm saying I have this cell, one cell, then I have this another. So they are going to keep cell junction, right? You can see the plasma membrane of both the plasma membrane cell, it's tightly, right? Let's just draw for better understanding. If I say I have two cells, and these cells, their plasma membrane is completely fused, so there will be no thing that can pass through cells or this plasma membrane. So tight junction performs function where it will not allow anything to pass the things or substances from one cell to the another, right? So the gap junction, talking about gap junction, it will allow the things to pass through. It's like a door between two rooms, right? You have two rooms and there is a common door between. Now everything, whether it's a small molecule, whether it's a large molecule, it can easily pass, right? So that's the beauty of a gap junction. It will allow the communication between two cells by the continuity, by allowing the continuity cytoplasm. Now its cytoplasm can move in this cell and its cytoplasm can move in this one. Talking about the adhering junction. Now what about these? Their main purpose is to adhere, right? That means it will it will keep the two cells together. It has some proteins which keep them connected like this. And it is also connecting two cells with the help of fibers like this. Okay, so it contains some protein fibers that helps to adhere both the cells so tightly so that they could not move, and they also helps to communicate with the fibers, not directly like gap junction, but indirectly through fibers. And these gap junction, like I said, these are two rooms to the door, and door is usually made up of wood. So these gap junction, what are they made up of? What is these are certain proteins, and what are these proteins? The connects. All right, so let's talk about them. Tight junction, it will not allow leakage of substances. Formed by complete fusion of plasma membrane. All right, next, the gap junction. Gap junction, it allows continuity of cytoplasm between, allows the continuity of cytoplasm cells, allowing the movement of ions, big or small. Next, adhering. It helps to adhere two cells together. It actually works as a cement. What is its function? Cementing. Helps in cementing. Right? So, what are these? These are fibers. And what is this green one? It's a plaque. It's made up of like a cement. All right. So, these are the three type of junction. So, it's a characteristic feature of epithelial cell because you will find it in all the epithelial. But these gap junction, you can also find this in cardiac muscle and the muscular. All right, guys. Moving further to the connective tissue. So, if someone asks you which is the most abundant, what do you want to say? That's a connective tissue. The most abundant and widely distributed. That means you will find it almost everywhere. If someone asks you what is its function, it helps to connect things. That means it will help to pack the organs as well. Helps to connect and pack tissues and organs. Next, like epithelial was avascular, this is the vascular. One more point, the comparison between epithelial and the connective. For that, we need to go back to the epithelium. If you can see here, we have written it's always present in the coverings and linings. That means the upper surface will always be free. That means it will not be covering by another. For example, if I see here, it's keratin. You cannot see any other of it. So we say that the epithelial tissue always have a free surface. Have a will not be covered by any other tissue above. Whereas if I'm talking about the connective tissue is lying just down here will be the muscle tissue and above is the epithelial tissue. So can you say it has a free surface? It never has a free surface, unlike that of the here. Connective tissue never has a free surface, unlike all right. Okay. So moving further, let's talk about what are the components of a connective tissue or what connective tissue is containing in, right? So if I talk about what connective tissue is made up of, it is made up of a base which is gel-like and you call it as a ground substance. And why it is gel-like? Because it contains some mucopolysaccharides. Because it contains mucopolysaccharide like hyaluronic acid, right? So for example, you have a jelly you eat, right? And that jelly contains small threads, you call it as fibers, and that jelly also contains gems, the Cadbury gems that you eat. So that gems, they are cells, and then small threads, they are fibers, and that jelly is a ground substance. So here we have the another components. What are those? These are the fibers, and then we have the cells. So in this section we'll talk about the cell first. What are the various cells it has? First is fibroblast. Yes, its name suggests fibroblasts are these cells that secretes the fibers. They secrete fibers. Talking about the next cell, that is a histiocyte. What are histiocytes? Histiocytes are the macrophages. Now, what are macrophages? Macrophages are the cells of your immune system. They are usually also present in the tissues. That you'll also learn in the nervous tissue. They have microglial cells. They're also macrophages. In the liver, we have Kupffer cell, and here we have histiocytes. The function of these macrophages is that they will engulf any microbe or any foreign particles that enter into your body, and then they will eat it. And that property is known as phagocytosis. So these cells, they shows phagocytosis. Right? So what is phagocytosis? Engulfing the microbes or any foreign particles, any dead cell, that's a phagocytosis. Then we have plasma cell. Now what are these plasma cell? They are also known as cartwheel cell because their nucleus appears like a cartwheel. Nucleus is like cartwheel. I think you all understand what is a cartwheel. Cartwheel, the one on the bullock cart, right? Like this, it appears like this. Now, what does this cell do? You must have heard of the cell B cells or B lymphocyte. They form antibodies. Similarly, in the tissues, these cells, they forms antibodies. Then we have another two types of cell. What are these? We have adipose cell, which are fat cells, which stores fat. They fat. And the last one is a mast cell. Mast cell are the cell which are responsible for allergies because they secrete three component. One is histamine, second is serotonin, and third is heparin. Right? What's the difference? Heparin is the anticoagulant. It will prevent the blood clotting. Histamine is a vasodilator. What is a vasodilator? Vaso means blood vessel and dilation means to expand the lumen. So whenever there is allergy, we need a lot of cells. We need a lot of cells so that they can come there and they can kill the whatever microbes are there. So there we need our blood flow to be more. So we expand the lumen of blood vessel. But serotonin works opposite to the histamine. It is a vasoconstrictor. Vasoconstrictor, it narrows the lumen. It narrows the lumen. Right? So these are the various type of cell that you will come across while studying this connective tissue. So let's talk about the another component, which are the fibers. So you will see there are three types of fibers. You can see this diagram. This is actually not a diagram, that's a microscopic photograph from a tissue culture. So you can see there are thick fibers here. These are the collagen fiber. So these collagen fibers, you can see these are unbranched. They're unbranched. You will not see branching here. They are made up of a protein which is collagen. They will be white in color and they will be in bundles. That's why they are so much thicker. For example, if I have one single stick or I have a bundle of sticks, the bundle of sticks will appear thicker and it will also will be having a lot of strength. So that's why collagen is, more, it has more strength and it is in bundles, so it appears thicker. Also, it is non-elastic, and since it is providing strength, so it will be present in all of the organs where you need a lot of strength, like bone and cartilage. Talking about the elastic fiber, you can see these fiber, these are elastic. These are elastic fibers, right? These fibers, they are made up of elastin protein. They're yellow in color. They are single. That's why they do not appear thicker, and they are elastic. That's why their name is elastic. Elastic, that means you can stretch it like an elastic. Fine. And they are branched and they are present in the blood vessels. Next, we have reticular fibers. They are made up of protein known as reticulin, which is a type of collagen only. So that's why I've written collagen. Since it is a type of collagen, the color will be white, they will appear single, and they are single, they will be very delicate. Like I've told you, if collagen is made up of bundles, bundles will have strength. If I have single protein collagen, that will be very much thinner and it will be having less strength. Then we have they are branched and they are present in liver as well as spleen. Now, what is this spleen? If this is your stomach, behind the stomach is present a small organ that is spleen, right? This is stomach. Spleen is a part of your immune system. It's a very important lymphatic organ, and it is the graveyard of RBC. This is the site of RBC destruction. What is RBC? Red blood cells. Let's talk about the classification part of your connective tissue. You have three types of connective: loose, dense, and specialized. Why it's a specialized? Because they're performing special function. In this category specialized one is divided into further two categories: endoskeleton, supportive or skeletal, and then fluid connective tissue. As the name suggests, it's fluid, so it will be fluid in nature, not the solid one, and its matrix will not be gel like. First of all, if I'm saying something is fluid, the matrix will not be gel like, and also they have no fibers, no gel like matrix, because they're fluid. Here comes two components: one is blood, another is lymph. Then we have endoskeleton, because it's a forming skeletal system of your body, and this is also supporting your body. So here we have bone and cartilage. What do we have? Bone and cartilage. Then comes the loose and the dense. How have we classified them? On the basis of how the components are present. If the components are loosely packed, it's loose. If the components they are densely packed, compact, then you will call it as dense. In dense, we have a regular and irregular, and in loose, we have areolar and adipose. Let's talk about them in detail. Areolar. In areolar, if I talk about which is the most abundant tissue in your body, that's connective tissue. And out of the connective tissue, which is the most abundant connective tissue, that's areolar. So that's come the first point, which says it's the most abundant and it contains all the components. This is very important diagram. Why? Because many a times the diagrammatic question can appear in your examination, and this is the most favorite one from this chapter. It contains all the cells. You can see fibroblasts, the one in the green, and from here the collagen fibers are emerging. You can see the macrophage. What is the name of the macrophage here in the connective tissue? Histiocyte. Also, you can see the mast cells here that secrete histamine, serotonin, and heparin, right? And also, you can see a lot of fibers down there. So, every type of fiber, every type of cell will be present in this tissue. Where are they present? They're present in subcutaneous layer, that is beneath the skin, around the muscles and nerve, because it helps in packing. What are the functions? First of all, it is present beneath the skin. So cutaneous means skin, and anything present beneath it is subcutaneous. So that's beneath skin. So for better understanding, I would like to draw a diagram of a rough diagram of a skin. First of all, skin is made up of epithelial tissue with the basement. You call this layer as epidermis. Below this is present a connective tissue and you call it as hypodermis. Sorry, dermis, not hypodermis, dermis. And beneath this is also present another connective tissue and you call this layer as hypodermis, right? So epidermis and dermis are collectively known as cutaneous layer because that's the upper part of the skin and this is a subcutaneous. All right. So the connective tissue, see, we'll move back to the epithelial ones first. Okay, so where's the epithelial tissue I have made in the first slide? All right. So here the tissue which this connective tissue is present. This is a part of a dermis and here the areolar is not present. Areolar will be present beneath this and that's your subcutaneous layer. That is your subcutaneous layer. All right. Moving further. Okay, it is also present in the submucosa of gut. So while I'll be teaching you the digestive system, I'll let you know the transverse section of the gut or the alimentary canal. The one of the layer of the gut or the alimentary canal is submucosa and that submucosa is containing areolar connective tissue. So what's the basic function of this connective tissue areolar? It helps in the organ, packing the organ, right? So that's your areolar. Next is adipose. So if you can see this diagram, let's move back. You can also find these cells. And what are these cells? These are adipose cells. Adipose cell. So that means we can say that some of the location of adipose and areolar tissue is same. Yes, absolutely correct. So adipose tissue will be containing which cells? Adipose cells or fat cell that will store the fat. And the fat performs three function. One is insulation. What is insulation? It prevent the heat loss. It prevent heat loss. How? If I have our body, this is our body, right? Our body is doing a lot of reactions and it is producing heat. Now there are possibility if we have no fat, we will lose all the heat. But if we have a layer of fat around us, the heat will remain inside and it will not move outside. So this is what insulation is. Also, it acts as energy reserve because whenever we need energy, the fat gets depleted and give you energy. Then it also helps in shock absorption. How? Because if I say it is present around your vital organs like kidneys, vital means important organs like your eyes, there it is helping you to protect. It is it is absorbing all the shock if somehow you fell off and you will never get your kidney broken, right? Because you have a lot of organs, some these bones protecting the kidney, plus kidney is also wrapped around by the adipose tissue, so that's why it also performs the function of shock absorption. You will find it in under the subcutaneous or beneath the skin, that subcutaneous layer, around the vital organs like kidney, and it is also present in your yellow bone marrow that we'll be discussing in the bone, where it is in bone. And it is also present in some animals as a thick fat layer. You call it as blubber. So what is blubber? Thick fat layer. It is present in the mammals, usually in the mammals which are living in cold environments like whale, like polar bear, because these are mammals and they're warm blooded and they have to maintain their constant body temperature. So for that, nature has given them the thick fat layer because they're not like human. They are not that conscious. They cannot wear clothes and they cannot use a room heater just like that. So that's why it is also given in the animals as a blubber. So that's about the adipose tissue. You can see this diagram. In this diagram, you will see a big fat droplet or fat storage area is here. So that's a big fat droplet. And here a small dot is a nucleus. So as you can see, in the adipose cell, you will see the nucleus at site, but the fat is present just centrally here. For example, if we have a normal cell, it will be having a centrally located nucleus. But in these cells, since there is a large fat droplet, so you know, they will kick this nucleus at one side. So it will appear like a ring. That's why you call these adipose cell as signet ring cell. Signating cell. Fine. So that's about the adipose tissue. Moving further to the next. Next, we have the dense connective. In dense connective, we have two types or two categories. One is regular, another is irregular. We have classified them on the basis of how the components and basically the fibers are arranged. First of all, in a dense connective tissue, the components are tightly packed. The components are tightly packed, as you can see, unlike that of loose. And also in the dense connective tissue, the fibers are more. The fibers are more, or they are abundant. So if I have a parallel arrangement of fibers, as you can see in this diagram, that will be the regular one. If I have scattered arrangement of fibers, then that will be the irregular one. So what's the location? There is one dense regular connective tissue known as ligament that connects your bone with bone. For example, if I have this bone and this is another bone, here in the joints, like this is one bone, this is another bone. So both the bones will be connected to each other via elastic component, that's the ligament. Now I have the muscle attached to the bone. Now the tendon will connect my muscle with the bone. So from bubbly, you can remember bubbly, right? Bone to bone ligament, right? Or just LBB. Otherwise, another one is simply understood. If you remember one, then another one is simpler for you, right? In irregular vein, the components are totally scattered and it is present in the dermis of skin. Right, let's go back here. So here in this one, which connective tissue is present? The dense irregular, the one which we have also drawn in the epithelial tissue. Fine. So this one is areolar, where adipose will also be present. In the dermis, we will see the dense irregular. All right. And here we have epithelial tissue. So this is how your skin is made up of. All right, guys, let's move further and talk about the cartilage. What about the cartilage? So cartilage, as comparison to bone, if I compare to the bone, it is soft and pliable. What is pliable? That you can easily bend it. You can easily bend it. Like your, if I say at the tip of the nose you have a cartilage, you can easily bend it like this. Yes, so that means it's soft and pliable. And if I talk about the cartilage, what it is made up of? First of all, it will contain a lot of collagen fibers. It will contain a lot of collagen fibers and it will be having cells. So for that, we need to make a picture of it. Let's see. If I say that's the matrix of the cartilage, that's the matrix of the cartilage, and this matrix will contain very special cell which are chondrocyte. So whenever you see the word chondro, that means cartilage. So these cells are chondrocyte. And the chondrocytes are present in the fluid filled spaces, that's lacunae. So whenever you see the chondrocyte, they will present in some fluid filled spaces. What is its name? Its name is lacunae. Fluid filled spaces. All right. If I talk about the matrix, this matrix will be having a protein known as chondrin protein. What protein? Chondrin. Also, the cartilage, they are avascular. That means they will not be having blood supply. That's why they took a lot of time to get healed. For example, if I say ear pinna has the cartilage, so we all girls we get piercing, but this is not healed up, right? Because it's avascular, and whenever you get piercing, you will not get the blood. That's the first reason, because that's avascular, and it is not, healing it, and it does not fill up easily even if I just remove my earrings for a day or two, because their healing power is very slow due to absence of the blood supply. So avascular, that means no, or scanty. Scanty means very, very small blood supply. Then what are the various types of cartilage we have? What are the various types we First is hyaline cartilage. Hyaline cartilage is just like your glass. Like a glass is clear, transparent, right? and it's fragile as well. It's fragile as well. So just like that, your cartilage is also fragile, and which is that? Which is that? That's hyaline. So it's transparent and fragile. So where do you find the hyaline cartilage? In between the joints, that's articular. Where it is present? In your joints. Like here and here, there is a joint, you will see the articular cartilage. You will find it in the larynx, the voice box. You will find it in the tracheal rings. And you will also find it in the costal cartilage. What is the costal cartilage? The cartilage present between sternum and ribs. Right? That's the costal one. Then we have the white fibrous. Then we have the white fibrous. Since its name is white and fibrous, what type of fibers it will be having? It will be having a lot of collagen fibers. So where do you find it? Pubic symphysis. What is pubic symphysis? It's between hip bones. Between hip bones. Ventrally. If I say this is your dorsal side and this is your ventral side, so ventrally, where your two hip bone joints, there is present a structure known as pubic symphysis, which is made up of white fibrous cartilage. And then we have intervertebral disc, the disc which is present between two vertebra of vertebral column, that contains white fibrous cartilage. You have to remember these because these are very important, right? They will be very much important because in the human physiology you will come across all of these time to time. And then we have yellow elastic. Definitely, what type of fiber it will be containing? It will be containing yellow fibers, the elastic fibers, right? So where do you find it? You will find it in the tip of the nose. That's why I was able to easily move it like that, because it's elastic. Ear pinna, where girls undergo piercing, and epiglottis. So if you see also the eustachian tube is present in the ear. If you see the one with the E are yellow elastic. E for elastic, ear pinna, epiglottis, eustachian tube. You just have to remember tip of the nose. Then hyaline contains Al, so all of these contains Al. So it's easier to remember, right? Yes, simple as that. Okay. So that's about the cartilage. Let's learn about the another one. Till that, then you can take a screenshot. You can write it. Otherwise, that will be pretty good and better. All right. Moving to the next is the bone. As comparison to the cartilage, bone is hard and non-pliable. Now, why is it hard and non-pliable? Because it contains inorganic part, and that's calcium salts. What calcium salts it has? Or it contains calcium phosphate, right? Like like the cartilage had the chondrin protein, it has the ossin protein. Which protein? Ossin protein in matrix. Now, what type of cells does a bone have? One is the immature cells known as osteoblast. Wherever the word blast comes, that means immature cell. They will form the bone. They will get mature and they will become the structural part of the bone, that is osteocyte, the mature cell. And the third category is osteoclast. These are bone destroying cells. Sometimes we need to remove the calcium out of the bone. So these osteoclasts will help to remove some components of the bone under the influence of certain hormones, right? That we'll be discussing in chemical control. So if I'm talking about these cells, like we have done, the chondrocytes were present in these spaces known as lacunae. You will find osteocytes also present in lacunae. But osteocytes have these projections or arms, but they are also present in spaces. And what do you call them as? You will call them as the lacunae. All right. Also, these osteocyte, these osteocytes, with the help of their arms or projection. What are these arms or projection? These osteocyte, with the help of their arms or projections, they will form these rings, and you call these rings as lamellae. What do you call them as? You will call them as laminae. For example, in the childhood, you supposed to ring, you generally, sorry, my you get stuck here. So during childhood, you guys used to play a game that was Ringa Ringa Roses, right? So what happened in Ringa Ringa Roses? All the kids, they used to hold their hands like this, and they make a circle, and then they move like that. Similarly, all these cells, for example, this is one osteocyte, and these are the arms. So these all arms of the cells, they are making the rings like that, and that ring is lamellae. So you will see there will be a lot of rings, concentric rings present like this. All will be containing osteocyte. Okay. What is this? This is osteocyte. This one ring is lamellae. This one ring is lamellae. But you will find there are a lot of lamellae present, or a lot of rings present in the concentric fashion. Right? And that forms one structural and functional unit of your bone. You must have seen this diagram in the NCERT as well. And this contains the inner Haversian canal in it, from which the blood vessels passes, right? So this is Haversian canal. These all are lamellae. This is one ring, this is another, this is the third one. These are lamellae. And in between them is a Haversian canal from which blood vessel passes. So this is how you know the bone also have blood vessels from which blood vessel passes. And this entire unit is known as Haversian system or osteome. And this is the structural and functional unit. So this is the one osteome. This is the one osteome or one Haversian system. You will find a number of them in your bone. Fine. So this is how your bone actually looks like. If you zoom into the bone, you will find these type of a lot of structural and functional units. So these are the structural and functional. Right? Okay. All right. So if I'm talking about a bone, you must have seen the bone like this in the cartoons, right? Like this. Usually a bone and a dog. You must have seen this, right? So if I talk about the long bones, you will see in the long bones, in these part, you will see the red bone marrow. This is the inner organic part. And in the center long portion, you will see the yellow bone marrow. Now, what are these? These all are tissues. What are these? These are tissues. This is a red bone marrow. So these are the tissues present inside the bones, and this is yellow bone marrow. This yellow bone marrow is actually nothing but it is the adipose tissue, and you will see in the center of the bone. So those who eat mutton, they must be knowing the central portion they eat, known as nulli, I guess, in Hindi, right? So that's the yellow bone marrow. It contains fat. And then the red bone marrow, it is a site of hemopoiesis. That means formation of blood cells, right? Site of hemopoiesis, that is formation of blood cells. So here your blood cells are formed. All right. So here the bone is so tightly packed. So the bone present in this region is compact bone. And in the upper part and the lower part where red bone marrow is present, the bone is it appears like a sponge, so you call it as a spongy bone. What do you call it as? You call it as right. So if someone asks you what are lamellae, you will say these are the concentric rings. And if someone asks you what are the lacunae, then you will say these are the empty space. All right. So I hope you understood this bone and cartilage. Let's move further and talk about the muscular tissue. All right, let's move to the next tissue and talk about it, and that is your muscular tissue. This is a very important tissue because it helps us to move. Yes, it helps us in locomotion and movement. So what is the origin of this tissue? M for mesoderm and M for is muscle. So as you can say, it's easier to learn. So the origin is mesodermal. Now, if I talk about the muscular tissue, what it is made up of, for example, the epithelial tissue was made up of epithelial cells. The connective tissue was made up of fibers, cells, and the matrix. What does the muscular tissue have? The muscular tissue, it is made up of muscle fibers, muscle fibers or muscle cell, muscle fibers or muscle cell. What are the properties? What are the properties your muscular tissue have? First of all, as I've told you, it helps you to move. So it has the property of contractibility. Yes, it has the property of contractibility. So it undergoes contraction and relaxation. This is how during contraction the muscle shortens, and relaxation, it comes to the original position. Then it also have the property of excitability. This is also the property of neurons. So neurons and muscles, they share this common property of excitability. That means they can respond to the impulse, right? Through them an electric current can pass through easily. Otherwise, it has the property of elasticity. What is this property? That you can stretch it like a rubber band. And extensibility, you can extend it, right? You can extend it like this, okay? So these are the four main properties of a muscular tissue. Let's talk about the types of muscles we have. What are the various types of muscles? We have skeletal muscle, we have smooth muscle, and then we have cardiac muscle. So there are three main types of muscles. So let's talk about them in detail one by one. So we'll start with the skeletal muscle. You must have heard of it. Skeletal muscle have other names as well. What are the other names? First of all, it is also known as voluntary muscle. You must have heard of it in the lower grades. Why you call it as a voluntary muscle? Because it is under your control. It is under our control or our will. For example, if I say the muscles of my arm, they are skeletal muscle, I can easily move it according to my will. It's in my control. So that is a skeletal muscle, right? So here it also have other names like striated or striped. So before discussing the striated and striped, let's talk about the skeletal. Why you call it as a skeletal muscle? Because most of your skeletal muscles, they are attached to your bones, and bones are the part of skeletal system. So they are attached to bones. Attached to bone. So first location, I have explained you that it is attached to the bone. What are the another locations? What are the another locations where you can find it? First, attached to the bone. Second, in your tongue. Yes, your tongue, you move it according to your will. You have a control on it. So all the muscles are skeletal muscles. Also, your pharynx. Pharynx also contains skeletal muscles. And upper 1/3 of your esophagus. What is esophagus? Your, yes, upper part of your gut, esophagus, your food pipe. Upper 1/3 of esophagus is also your skeletal muscles. Talking about your abdominal wall, that is also skeletal. Now, what abdominal wall I'm talking about? I'm talking about this, your six packs and your back. These all are abdominal. This all area is abdomen, and the muscles there is the abdominal wall muscles, which are skeletal muscles. Then we have striated or striped. Why you call it a striated or striped? Striated or striped means there are certain lines or stripes present on the muscle. If I draw the diagram of this muscle, the muscle is cylindrical in shape. The muscle fiber, this is a muscle fiber. As I've told you, the muscles are made up of number of cells. You also call them as muscle fiber. So this entire cluster is a muscle, and this only single cell is a muscle fiber. So you will see the alternative light and dark bands here like this. So they are present in the form of striation. So you call it as a striated. They also appears like a stripe. So you also call it as the striped muscle. These are alternative light and dark bands. That means one after the another. One light, then dark, one light, then dark. And these dark and light bands are formed by the contractile proteins that helps the muscle to contract, and that are actin and myosin, right? So why you call it as striated or striped? Because of presence of alternative dark and light bands. Dark and light bands. So if someone asks you what the dark band is made up of, the dark band is made up of the myosin and light is made up of actin. And what are these? These are contractile proteins. These both are contractile proteins. Right? Then, if I'm talking about the shape of the muscle, what's the shape of the muscle? It's cylindrical. It's cylindrical. Let's talk about its nucleus. The condition in this muscle is multinucleated. That means there are a lot of nucleus. Since it is formed by the fusion of number of cells, so you also call it a syncytium. Synctium. You also studied in the botany. So here you will see a lot of nucleus, and these nucleus, you will find them in the periphery. Now what is a periphery? For example, you must be sitting somewhere in your room, right? So if I say just in the center of a room a nucleus is present, this is what occurs, or this is what happens in a normal cell. But imagine this particular room is a skeletal muscle cell, and there are a lot of nucleus, but the nucleus are present either around the walls or the roof or the floor, right? But there is no nucleus inside or in the center, but the nucleus are present at the periphery, on the walls or the floor or the roof of your room. So this is what we say, it shows multinucleated condition, multinucleated condition, and nucleus present at periphery, at periphery, right? All right, what about the other things this muscle have? This has a lot of mitochondria, a lot of glycogen. Why? Because it has to work a lot, yeah, right? It has to work a lot. That's why it has a lot of mitochondria, it has a lot of glycogen. Another thing it has is sarcoplasmic reticulum. What is sarcoplasmic reticulum? This is the modified smooth endoplasmic reticulum. This is the modified smooth endoplasmic reticulum that stores calcium ions. That stores calcium ions. Right? Since it is a skeletal muscle and it is not an ordinary cell, so it's sarcolemma. It has sarcolemma. What is sarcolemma? It is just the plasma membrane or a plasma lemma that contains certain glycoprotein. So wherever the word sarcolemma comes, you just directly have to say it's the plasma membrane of skeletal muscle. So what is this? This is the sarcolemma. This is also the sarcolemma. What is sarcolemma? The plasma membrane. But it's little modified because it contains certain glycoprotein. Then what will be the cytoplasm known as? Sarcoplasm. So what is sarcoplasm? Cytoplasm of your muscle, skeletal muscle, right? Now these muscles, they have a structural and functional unit which is formed by the alternative light and dark bands, and that is known as sarcomere. So sarcomere is also present. Sarcomere present. It is a structural and functional unit. Structural and functional unit. And this we will be discussing in detail in the chapter locomotion and movement. Right? Sarcomere is formed by the alternative light and dark bands. So for example, if this is a light band, this is a dark band, and there will be a very small unit that you will call it as a sarcomere. Also, this muscle gets fatigue. What is fatigue? That means it gets tired. It gets fatigue. Why? Because sometimes when you run so much, like your leg muscles, they get fatigue. You get soreness in the muscle, you get cramps in the muscle. That is known as a muscle fatigue, and that occurs due to accumulation of lactic acid. What happen is when the muscles are respiring anaerobically, you also know that whenever the muscle respires anaerobically in the absence of oxygen, that glucose is broken down into the pyruvic acid, and then pyruvic acid gets broken down into the lactic acid when there is no oxygen present. But if the oxygen is there, definitely the pyruvic acid will undergo the Krebs cycle and all, and it will ultimately form energy and form CO2 and water. But when there is no oxygen, it gets fatigued due to accumulation of lactic acid, because when there is no oxygen, the glucose gets broken down into lactic acid. Now, when is lactic acid formed? When glucose get broken down, it forms lactic acid when there is no oxygen. All right. So that's all about the skeleton muscle. Let's move further and talk about the smooth muscle. You also call smooth muscle as a visceral muscle, and the unstriped or non-striated. Let's see. Okay. So why you call it as a visceral muscle? Because it is present. It is present in visceral organs. Now, what are your visceral organs? Visceral organs are usually organs that are hollow inside. For example, it is present in your gut, it is present in your urinary bladder, it is also present in the uterus. So, what is the location? The gut, urinary bladder, uterus, blood vessels, etc. Right here, you will find your smooth muscle. Now, why you call it as a smooth or unstriped or non striated? If you look at the fiber of your smooth muscle, this is one smooth muscle cell. This is one smooth muscle cell. You can see it has quite different shape as comparison to the skeletal muscle. The skeletal muscle was cylindrical and multinucleated, but this one is uninucleated. Let's just move back. As you can see here, this is a nucleus. The nucleus. Their shape is cylindrical and there are a lot of nucleus present at the periphery. And if I compare it with the smooth muscle, what is its shape? The shape is fusiform. What is a fusiform shape? Whenever anything is thicker from the middle portion and from the ends, it's tapering. These are tapering ends. You call that shape as the fusiform. Also, it has only one nucleus. It has only one nucleus. So you call it as the condition is uninucleated. Uni means one. Also, there are no light and dark bands. No light and dark bands present. So you call it as it's unstriped or non-striated. And the appearance is smooth because you cannot see any band. There is no roughness. So that's why you call it as the smooth or unstriped or non-striated, because alternative light and dark bands are absent. Light and dark bands are absent. Right? So does that means it do not have any contractile proteins? It do have. Now what's the story? If I say that's a skeletal muscle and these actin and myosin, or these alternative and light and dark bands, these are present in the cylindrical structure known as myofibril. So we say that these structures are present in the muscle. These are myofibril. So this is one myofibril, right? These myofibrils, how are they present? They are transversely present. And these contains alternative light and dark bands. But if I say these are not present transversely, but these are present longitudinally. So they cannot, because they are present longitudinally, they cannot make the alternative light and dark bands. So it's the arrangement that makes these alternative light and dark bands. If there are no alternative light and dark bands, then there will be not any sarcomere as well, right? So we say there is no alternative light and dark band, so there will be no sarcomere. As I've told you, due to these alternative light and dark bands, the structural and functional unit is formed there, which you call it as a sarcomere. But here myofibrils longitudinally present. But there the myofibrils were transversely present, so they were showing that arrangement. All right. Next, talking about the smooth muscle, it will not be containing any sarcomere and there will also not be any sarcolemma as well. It will be having the normal plasma membrane. Also, it has low glycogen and low mitochondria. Low mitochondria. If I compare with the skeletal muscle, it do not get fatigue. It do not get fatigue. Not get fatigue. As it is present in the gut. Have your gut or your stomach ever said to you that I'm tired? Let's not eat some food. I'm tired. I'm not going to do anything. No, not like that. So that's why I say the smooth muscle, they do not get fatigue, right? And some smooth muscles also have gap junctions. Like I have told you in the gap junctions as well, they have gap junctions, right? So that's about the smooth muscle. Next, let's talk about the cardiac muscle. The cardiac muscle is a muscle that is present in the heart. Present in the heart. It has some characters of the skeletal muscle. It has some characters of the cardiac muscle. So let's talk about them. So if I say about the skeletal muscle, like skeletal muscle, it is cylindrical in shape. It is cylindrical in shape. Also, like skeletal muscle, it has sarcomere and sarcolemma because it has alternative light and dark bands. As I've told you, due to alternative light and dark bands, there is a formation of sarcomere. So that's it. If it has sarcomere, that means it must be having the alternative light and dark bands. Alternative light and dark bands. All right. Now, one character, which is also of your smooth muscle. That smooth muscle is involuntary. That means it is not under your control. So, have you ever tried to control your peristalsis, that the gut movement? No, never. So, that's the sharing portion, that it is also involuntary. It is not under your control. Fine. Otherwise, if I talk about the cardiac muscle, it has a very, very important and good character that is exclusively for the cardiac muscle. That is, it is autorhythmic. What is autorhythmic? That means it can generate its own action potential. It can generate its own action potential. You also use the word auto excitable here. That means it do not need any neuron to tell it that let's contract. So that's why we say our heart contracts itself, because it is auto excitable. It is not under the control of the neuron to initiate its heartbeat, right? So that's why you must have heard of a thing that if you keep the human heart out from the body, it will keep on beating for some time interval, because it does not need the electrical impulse from the neurons. It can generate its own, right? Also, it contains a lot of mitochondria because it has to work all the day, right? And it has a lot of glycogen. It also do not get fatigue. It do not get fatigue. So let's draw the structure of this muscle. As I've told you, it's cylindrical, but the fun part is it's branched. Yes, it's branched. So this muscle is branched. So have I ever discussed with you that the skeletal or smooth is branch? No. So that's an exclusive feature of your cardiac muscle. This muscle is branched. In between the cell, if I say this is one cardiac muscle fiber, this is another cardiac muscle fiber or cell. So you will see that there are bridges present. So you call it as oblique bridges. Oblique bridges, which is connecting one fiber with the another. Also, you can see there is only one nucleus, so the condition is uninucleated here. And in between two cells, there is present some disc-like structure, and you call it as intercalated disc. Now, what does these disc contains? These disc, they contain gap junction and adhering junction. Now, why we have a gap junction here in the smooth muscle and in the cardiac muscle? Because if the electrical impulse has been arrived in this muscle fiber, it can easily transfer the information down here to the another cell. So that's why we have gap junction, because we have discussed the gap junctions. There can be easy movement or passage of the ions, right? So ions are like, whenever the ion move, there will be an electrical impulse. So if the one ion is moving from here to here, it is going to transfer the impulse into this one. Also adhering junction, because while contraction, maybe this can be a possibility that these cells get separate up. So we don't want these cells to get separate up because there is some arrangement. So that's why there are adhering junction that will provide the function of cementing, and they will adhere both the cells so finely that they will not move anywhere. So that's about the cardiac muscles. Moving further to the nervous tissue. Let's talk about the nervous tissue. So if I talk about the nervous tissue, what's its origin? Its origin is, yes, boys and girls, you know that better than me now. Origin is ectoderm. The origin is ectoderm. But again, there is one exception. With an exception, that is microglial cells. And these cells, they are mesodermal in origin. These are mesodermal in origin. So if I talk about the nervous tissue, what type of cells does it contains? What are the various cells? We'll get two type of cells. One are the neurons and another are the neuroglial cells. Neuroglial cells. Now what's the difference? These cells, neurons, they are excitable cells. That means they can generate the action potential. They can generate the electrical impulse, right? But here, neuroglial cells, these are non-excitable. But these cells, they can divide. And you must have heard ever in your life, if you have ever heard it, that yes, the neurons, they cannot divide. So whenever there is a brain tumor, you must have heard of a brain tumor, cancer. Then what is the reason of brain tumor? What is a tumor? It's the uncontrolled division of cell. So in brain tumor, the neurons are not responsible for the tumor. What are responsible? The glial cells. So these are the one that actually gets uncontrolled divisions, and they turn into the tumor. So they divides uncontrollable and forms tumor, right? But if these are excitable, they are doing a very crucial role. That means they are sending the impulse and message to the entire body, to the entire organs. They're coordinating the body activities. So they will be the structural and functional unit. So these are the structural and functional unit. All right. So let's talk about the neurons now. So let's draw the structure of a neuron. The body of a neuron is divided into two parts. How many parts? Two parts. You can see here. So the body of the neuron is divided into two parts. Let's just discuss. The upper part, this one, is the cyton or the cell body. Whereas the lower part, this one, is the axon. You can see in the cell body, you can easily find here nucleus, because that's the main portion. And these branching, you can see here, these are dendrites. What are they? These are dendrites. Dendrites are branch structure. They are tapering. And they are afferent. What is afferent? Afferent means towards. Afferent means towards. They will be bringing the impulse or the message from the other neuron or the nerve cells, right? Here in the center, as I've told you, what is this? This is the nucleus. You will find small granules like particles in the cell body, and in fact in the dendrites as well. What are these? These are Nissl granules. Nissl granules, if someone asks you, are they present in the axon? No, they are just present in the cell body and the dendrites. These Nissl granules, they contains ribosomes and rough endoplasmic reticulum. So now you know what they do. They help in protein synthesis. Because wherever the word ribosomes appears, that means they are the site of protein synthesis, right? Then down here we have the axon, and sometimes the axon give branching at the 90 degrees, and this branch you call it as axon collateral, right? So that's the plasma membrane of the axon. You call it as axolemma. And this axon is wrapped around by some cells, and these cells are known as Schwann cells. What do you call them as? Schwann cells. Schwann cells. So these are Schwann cells. These cells secrete white color lipid. You call it as a myelin sheath. What do you call it as? Myelin sheath. So in the some places you will see the axon is wrapped around by Schwann cell, and there is also presence of a lipid. You call it as myelin sheath. So what is this? This is myelin sheath. Myelin sheath is a lipid, so it acts as the electrical insulator. So wherever the myelin sheath is present, there will be no passage of the electric current. And there are then places where the myelin sheath is absent. You call it as node of Ranvier, right? All right. So myelin sheath acts as an insulator. I think you all understand about physics. What is an insulator? From which the you cannot pass the electric current. And places where is no myelin sheath, that is the node of Ranvier. Okay, that's not Ranveer Singh. That's Ranveer. All right. Then you have these end bulbs or synaptic knobs in the end portion. These end bulbs or synaptic knobs, they contain small vesicles. Vesicles are what? They are sac-like structures. These small vesicles, they contain a chemical known as neurotransmitter, and the name of these vesicles are synaptic vesicles. And what do they contain? They contain neurotransmitter. Now, what are the neurotransmitters? Neurotransmitters are the chemicals. These are the chemicals that helps to transfer the information from one neuron to the another. How? Let's see. If I say there is the another neuron present just right here, right? They are not in the direct contact. As you can see, there is a space left in between both these sides, right? In both of them, there is a space present. Here is a space present, right? Yes. So this functional contact, not the physical contact, is known as a synapse. What do you call it as? You call it as synapse. From this synapse, there is the transfer of information. That means, for example, this neuron has an electrical impulse. So we need to generate the electrical impulse into the second neuron. This is neuron 2, this is neuron 1. So how they're going to transfer the information? They're going to transfer the information by giving it the chemicals that are neurotransmitters. All right. So what is a synapse? It's a functional contact between two neurons. It's a functional contact between two. All right. And it can also be between a neuron and a muscle. When it is between a neuron and a muscle, you usually use the word neuromuscular junction. Use the word neuromuscular junction. I hope that's pretty clear to everyone. What's a synapse? In synapse, there is no physical contact. They're not touching each other. Rather, they are present away from each other, just like this. How this one is going to send the information to this one? By giving it the chemicals, and these are neurotransmitter. For example, if this is neurotransmitter, this has a neurotransmitter, it will release it, and this will get the information. All right. So this was about the structure of the neuron. Let's talk about the neuroglial cells. What about the neuroglial cell? If I say more than 50% of the cells are neuroglial cells, then yes, that's true. So more than 50% of the nervous tissue are your neuroglial cells. So the usually function is to protect the neurons, to insulate them, to repair them. So let's see what are the various types of neuroglial cells we have. We have astrocytes. Astrocytes are the cells which are star-like. Aster means star, right? Second, we have the Schwann cell that we have discussed that secrete myelin sheath. Then we have oligodendrocytes. And then we have the microglial cell. So apart from a neuron, you will find very small cell. For example, if this is your neuron, right? This is neuron. Around the neuron, you will find very small cells, and these are neuroglial cells. They have different categories. So we are talking about these categories now. What are the different types of neuroglial cell? We have astrocyte. These are star-like. These are just star-like, like this. Okay, what is the function? They will repair the neuron. They will insulate the neuron. They will form the blood-brain barrier. blood brain barrier. Now, what is the blood brain barrier? Your brain contains a lot of tissues, right? It contains a lot of neurons, and neurons, they are so much important, and once they are broken down, you cannot make it new because they cannot divide. So your blood has to go into the brain. Why? Because blood contains nutrients, it contains oxygen, right? So you cannot give the entire blood to the brain because in the blood a lot of harmful substances may be present. So we generate a barrier in between two. So the only few particles can pass through these barriers and can enter into the brain, so that no harmful substances of the blood can enter into your brain, right? So that's why nature have given a barrier between your blood vessel and your brain. For example, this is your brain, right? And this is the blood vessel around the brain. So in between these two is present here like this blood brain barrier. Is this blood brain barrier, and this is formed by the astrocytes, right? Along with the astrocyte is also present another neuroglial cell that are ependymal cells. So they form blood brain barrier with ependymal cell, and ependymal cell is another category. It's the another category of neuroglial cell. Okay. Then we have Schwann cell. It forms myelin sheath. What does it forms? It forms myelin sheath, right? What about oligodendrocyte? They also forms myelin sheath. Then what is the difference? Oligodendrocyte forms myelin sheath in central nervous system, that is brain and spinal cord, and it forms myelin sheath in PNS, peripheral nervous system. Talking about microglial cells, they are mesodermal in origin and they are like macrophages. So as we have discussed, the macrophages of the connective tissues, they were histiocytes. What are macrophage? They actually engulf the microbe or the another particles. They can undergo phagocytosis. So these are the one which are the which are actually protecting the entire nervous tissue, so that if any microbe or anything comes, they can easily engulf it. So that's all about the nervous tissue. Now it's time to solve some question. We have covered all the four tissues. Now it's the time to see how much do you learn. Let's see. Alright, so this was it about the theoretical part. It's time to solve some of the previous year questions. So why previous year questions? Because they are very much important from the examination point of view, whatever examination you are giving. If you're preparing for NEET, go for the PYQs of NEET. If you're preparing for KVPY, go for the previous year questions of KVPY. That gives you merely an idea about what is relevant and what is irrelevant. And that's really a must thing. You should not read which is not relevant according to the examination. So let's get started. First question, as you can see, the first question is diagrammatic. So this diagram we have already done in a tissue. So this is how you get to know that how diagram can be very much important, right? So let's read it. Given below is a diagrammatic sketch of a certain type of connective tissue. Identify the parts labeled A, B, C and D and select the right option about them. Part A, B and C and D are given and they are labeled as well in the diagram, and you have to identify which are these. So A option says the part A is macrophage, B is fibroblast, C is collagen fibers and D is mast cells. And B option says A is mast cell, B is macrophage, C is fibroblast and D are collagen fibers. And C says A is macrophage, B is collagen fibers, C is fibroblast and D is mast cells, and D says the A is mast cell, B is collagen fibers, C is fibroblast and D are macrophages. So whenever these type of question arise in exam, do not go for the options first. First of all, go and write the labelings, right? So for example, if you know A is a macrophage, that's pretty clear, right? And what's B? B is your fibroblast and C are your collagen fibers. Whereas what is D? D is your mast cell, right? So now straight away go to the option and mark it. What is A? Is collagen fiber. What is B? B is your fibroblast, right? A is macrophage. You can see where is A. In option A, in part A, we have A. B is fibroblast and C are collagen fiber and D is a mast cell. So answer is A. That matches your option A, right? So it's quite simpler. So whenever you read the options first, you actually got confused, right? So you don't need to confuse the options. What is the purpose of multiple choice? The purpose of multiple choice question is to confuse you with the other options. But if you are sure that you have read this previously and you have read it very carefully and you have learned it as well. There is no if and but, you just directly go to it and be very sure and then go to the option part, right? Simple. All right, next. The supportive skeletal structure in the human external ears and in the nose tip are the examples of. Now, it is talking about the supportive. What two organs or what two tissues provide you support? One is a bone, another is a cartilage. And which is present in the tip of the nose? That's cartilage. So, answer is A. I think that's simpler. Yes, guys. I think you, if all these questions are correct, that means you have learned the chapter very nicely. All right, next question. Which type of tissue correctly matches with its location? So there is a tissue, another is a location. A. Cuboidal epithelium lining of stomach. B. Smooth muscles wall of the intestine. Then areolar connective tissue or the areolar tissue, tendons, and transitional epithelium in the tip of the nose. You have to find which of these following is a correct one, right? So another point. Sometimes you do not read this word correctly, right? Or sometimes you read the incorrect as correct, or sometimes it's written not correct, or sometimes it's written not incorrect. So first of all, you have to understand what the question is asking you. Based upon that, you can solve the question then. So here the tissue cuboidal one is not present in the stomach. In stomach we have the which one? Columnar, right? Whereas cuboidal, brush bordered cuboidal is present in your PCT. Smooth muscles, the smooth muscles, yes, they are present in the wall of intestine. Whereas the areolar tissue is not tendon. Tendon is your dense regular, right? Now, transitional epithelium is also known as urothelium, that is present in your renal calyces in your urinary system. Whereas at the tip of the nose, what do you have? You have cartilage, right? So likewise, the answer is B. I think now it's pretty clear that how all these tissues can arise in one single question. Yes, and that's the game. All right, next. The function of the gap junction is to stop substances from leaking across the tissue, performing cementing to keep neighboring cells together, facilitate communication between adjoining cells by connecting the cytoplasm for rapid transfer of ions, small molecules, and some large molecules, and separate two cells from each other. Now, that's a question from cell junctions. That's quite important because the question that came is just simply the definition from the NCRTs, but you should know each and every word very carefully. So, which junction stop the substances from leaking? That's tight junction, right? Performing cementing is the function of adhering junction, whereas facilitates a communication between two cells. There are two cells, and which junction allow the cytoplasm to move here and here? That's the gap junction. So, C is the correct answer. The one that separates two cells from each other is again tight because that maintains the identity of two cells, right? So, answer will be C. Choose a correctly matched pair: tendon, specialized connective tissue; adipose, dense connective tissue; areolar, loose; and cartilage, loose. So, tendon is not a specialized, it's a dense connective tissue. Adipose is a loose, whereas areolar, it's correct, it's loose, and cartilage, it's a specialized one. So, what will be the answer? The answer will be C, right? All right, next question, guys. Match the following cell structure with its characteristic features. Select the correct option from the following. Alright, so you just have to do match the following. It is again, it's a one of a recent question in the recent years it has arrived. And now you can see here, again, it's a question of cell junctions. So see how much important the cell junctions are. And if you have noticed, if you'll read this question as well, you will see the only definition is given. They are not asking so much important or so much of a high standard from this question. It's quite simpler. It's NCRT based. So you need to just learn them very carefully. Yes, you do have to learn the definition because if you will read this, tight junction. The second is cement neighboring cells together. That's the exact word they have used in NCRT. Adhering junction, transmit information through chemical to another. Gap junction, establish a barrier to prevent leakage of fluid across the epithelial cells. And then synaptic, cytoplasm channel to facilitate communication between the adjacent cells. So let's just match them up first. So, first of all, guys, Tight junction. What is the function of tight junction? It prevents the leakage across the epithelial cell because tight is present exclusively in epithelial cell, whereas gap junctions are also present in your cardiac muscle and smooth muscle and obviously in the epithelial cells. Then we have a deadening junction. Its function is cementing. Gap junction allow the continuity. Now, synaptic junction, they are not the feature of epitheliums. They are the feature of neurons and they allow the communication between the two cells. So what do we get here? We get A third, then B 1, then C 4 and D 2. If you write this in examination, you can never get confused and match the following. You know, one mistake that a student does in the examination when the question of a match the following arises, that is they get confused in these option and somehow they know the correct answer but they mark the incorrect in the OMR. So what you have to do is first of all match them up, write this in this manner and then mark the answer, right? For example, A third, B1, C4 and D2. Now you are sure an answer is D, right? If you just does it in your mind only, you are going to get confused, and because at that time, you know, your heartbeat is already so much up, but you don't need to do that's just exam and you have prepared well. So then don't just get stressed in the exam, because like if you are stressed in the exam, you can do these silly mistake like that, right? So don't just get confused in that. Fine. All right. Okay, next. The ciliated epithelial cells are required to move particles or mucus in a specific direction. In humans, these cells are mainly present in. So they're talking about ciliated epithelium. So where have we done the ciliated? We have done the ciliated in bile duct and bronchioles. We have done in the bronchioles, but not in the bile duct. We have done in the fallopian tubes, but not in a pancreatic duct, because in the pancreatic duct we have cuboidal, right? Next, the Eustachian tube, it contains cartilage, whereas salivary duct also contains cuboidal. Whereas bronchioles and fallopian tube is the correct answer, because here you want to move some things in a specific direction. So they're asking, or I think they have specified as well, like mucus. They have given one example. So in bronchioles, we move the mucus, whereas in the fallopian tubes, we move the ova or egg of the female, right? So answer is D. Fine. Next. Nasal bodies are mainly composed of. So what are nasal bodies made up of? They are made up of rough endoplasmic reticulum and ribosome. Where have we done? When we have made the structure of the neuron, if you remember, in the structure of neuron, we have drawn some small bodies which are nasal bodies or granules. And what do they contain? In the cyton structure, we have drawn these structure. They contain rough endoplasmic reticulum and ribosome, and both these are, whenever you hear the word ribosome, that are the site of protein synthesis. So the answer will be they contain free ribosome and RER. Answer is C, right? These are not only present in the cyton, but they are also present in the extensions of cyton, which are dendrites, but they are absent in the axon. Fine. Next, smooth muscles are, now here are the features written. You have to find out the correct features of smooth muscle. They are involuntary, very true. They are also fusiform. What is fusiform? Of this shape. And they do not get any or have any striations. The first is absolutely correct. Let's see what's wrong with the other three. They are not voluntary, they are involuntary, right? Next, they are not cylindrical. They are also not multinucleate. They are also not cylindrical. They are involuntary, they are not cylindrical, and they are not striated. They are also not voluntary. They are spindle shape. The fusiform, another name for fusiform is spindle shape, right? You understand what spindle, right? Have you ever seen spindle? The one that's thicker from the middle and it's very tapering from the outer ends. And then it's uninucleated, true, but the answer with all the correct features will be the answer, and that's answer A. Fine. So from the muscle, these type of questions are like so much expected to come. So you must read all the feature of the muscles very carefully. Next. All right. So time is precious. Utilize it carefully. All right. So we have done some of the previous year question. I'll meet you in the next class where we'll study the another chapter. Till then, you just what you have to do is whatever chapters have been covered in zoology by Vicky sir or by me, you just have to finish them up first. Read the NCRT and lock it up. Lock it up means you know it very well. You have done it so nicely and so good. I'll meet you in the next class. Till then, let me know what have you studied and how much you have completed in the comment sections. Bye bye.